Assalamu alaikum everyone. I welcome you back to this MBB-505 Techniques in Molecular Biology 1. Today we will be studying SDS page, that is in the form of electrophoresis, gel electrophoresis. The only difference is that when we do agarose gel electrophoresis, we use agarose there as a matrix. And when we use SDS page, we use polyacrylamide gel here. The constants are different, the principle is the same, though mostly if we run a protein sample, we usually use SDS page. And when we run a DNA sample, we use agarose gel electrophoresis. However, DNA can also be run on page, like sequencing gels or small molecular weight DNAs, because the resolution of page is better than resolution of an agarose gel. Okay, so SDS stands for sodium decyl sulfate. Okay, before we continue, I request everyone kindly switch off their mics. If you have a question during my lecture, you can raise your hand and I'll come to know that you want to ask something. A page stands for polyacrylamide gel electrophoresis. Okay, so we usually in our lab run proteins in this SDS page, and while running an agarose gel, we have to, it's very easy to prepare the gel and to prepare the DNA. However, in this case, it's a little bit tedious because there are various constituents. And in this SDS, we usually run two gels. Okay, one we call as the stacking gel and another we call as resolving gel. So if we draw it here, so this gel has two parts. The upper part we call as stacking gel. And the lower one we call as resolving gel. The pH of this stacking gel is 6.8, while as the pH of the resolving gel is 8.8. And the pH of the buffer, that is usually tris glycine, is 8.3. Why there is a difference in the pH, I'll be explaining that. Okay, so this is this gel, and when we make an agarose gel, the cast is open and we pour the gel in and let it solidify. However, when we make a polyacrylamide gel. We take 2 glass sheets, okay, and we put them together with the help of a small spacer. Okay, they have spacer on 3 sides so that when we join these glass plates, they will have a little space in between. Okay, in which we'll pour our gels. Fine. So why we the question arises why we run or why we make this polyacrylamide gel in a glass chamber, okay, thin glass chamber. However, we make agarose gel openly because the main constant of the gel is acrylamide and bisacrylamide. They do not polymerize in presence of oxygen, so you have to take out all the oxygen. That's why we make it in between 2 glass plates. And once we pour the gel in this, we seal it with isopropanol. So what are the things that we require in this? So number one, we have to make this stacking gel. Okay, the constants of stacking gel are you need tris HCl. Okay, this should be around 0.5 molar. Okay, now what we do in lab, we make a 5 molar or a 2 molar solution of tris chloride, and then using the same formula working by Stark that we studied in our previous lecture, we can calculate that if you are making a 5 ml gel, how much tris chloride will be required. Then you use SDS, 10%. Then you need acrylamide and bisacrylamide in the ratio of 29:1. So the major proportion of this acrylamide gel is acrylamide. However, you have a small 29:1 ratio, bisacrylamide you'll require. Okay, then you require APS, ammonium persulfate. Okay, that should be around 10%. And you require timid. Okay, this is a pre-made, you required 5 microliters of this. So as an agarose gel, the matrix is made by agarose. However, in the SDS, the matrix is made by acrylamide and bisacrylamide. Okay, they cannot form bonds and they cannot form matrix on their own. That's why we add APS, ammonium persulfide. It provides free radicals that helps in the bonding of these acrylamide molecules and bisacrylamide molecules. Okay, so this APS provides free radicals, and timid acts as a catalyst. It acts as a catalyst and it helps in the formation of this gel. It catalyzes this whole reaction, the formation of this matrix, and joins acrylamide and bisacrylamide with the help of APS. Fine. And tris HCl maintains the pH. It's a buffer, and you know what is the function of a buffer. Okay, it undoes any change that happens in the medium or because of its properties. So these are the requirements of the stacking gel. Okay. This is the stacking gel. And the requirements for your resolving gel are also called as separating gel. Besides this, we make the final volume to 5 ml with distilled water, okay? That you should remember. So now in case of resolving gel or separating gel, you require 1.5 molar tris HCl, okay? You require 10% SDS, okay? You require acrylamide bisaccharides 29:1, you require 10%, and you require timid. Here, timid, you require 10 microliters. Okay, but once you are making the gel, first you will make the resolving gel, because when you pour it in between the glass slides, first your resolving gel will be up to more than the half of the gel. Okay, if this is these are the gel plates. Okay, so resolving gel will make up till here. Okay, more than half. Half is here, so more than half. Okay. Then once we add this, we do not want that there should be any oxygen bubbles or oxygen in contact with the gel. Plus, when we pour the gel, there is this bubbling or froth formation. Okay, that's not good for a good gel, okay, for a good experiment. So we need to remove it. How do we do it? We add, say, 50 microliters of isopropanol. Once we pour the isopropanol, these bubbles disappear. Okay, isopropanol floats on the top. Okay, it changes the surface tension, due to which bubbles will disappear, and it forms a thin layer over your resolving gel so that it can keep the oxygen out, because in presence of oxygen the gels do not solidify. Fine. After say 10 to 20 minutes, so you mix all of these. At the end you add timid. Okay, shake it and pour it in the gel. So after 10 to 20 minutes this will solidify. How will you know? You'll take out this plate and just tilt it. You'll see that the ethanol will come out, but the gel will be there. So after that, you will, with the help of a wash bottle, you will wash this isopropanol out. Okay, 2-3 times. You will put distilled water in here. Okay, rinse it and throw distilled water. So you have to ensure that you remove all the isopropanol. Otherwise, if there is a mild amount of isopropanol between the gels, they will lose the contact. Okay, they should be in close contact with each other, the dissolvent and the stacking gels. Once done, then we come to your stacking gel, mix them, add timid, and pour it over this gel. So it will be like this. This is resolving, this is stacking. As soon as you pour the gel in there, you will put on comb, like we have the comb in agarose gel. You will put a comb also here. In these, so it will make wells when the gel will solidify, like yesterday. Okay, so these wells, when the gel solidifies, you take out the comb, so you will have these protrusions or wells in which you will load your protein samples. Any questions till now? No? Okay. like agarose gel, this is a little bit tricky and tedious because you have to prepare protein samples before you load them in the gel. Yesterday we saw that we had that loading dye. In this case, we have the loading dye that's called a Lamely buffer. Okay, we have to mix our samples, protein samples, with this Lamely buffer and heat it for 10 minutes or boil it for 10 minutes. So the constants of this Lamely buffer are, you have 4% SDS, you have 10% beta mercapto ethanol, it's a reducing agent. Then you have 0.004% bromophenol blue, the same bromophenol blue that we use in agarose gels. Okay, and 0.125 molar tris HCl. So these are the 4 constants plus water, constants of your Lamely buffer or the loading dye. So this function of bromophenol blue, you know, it tracks the gel, and we'll be able to see that how much our gel has run. Okay. SDS helps in the denaturation of the proteins because in case of DNA, we know that they have negative charge due to phosphate. Okay, and the charge is equally distributed throughout the DNA strands, and this charge helps in the running of the DNA samples in an agarose gel. Okay, in case of proteins are, some are amphoteric, some are negatively charged, some are positively charged, some do not have a charge. Okay, so to avoid this, in their native forms, proteins will not be able to run in a gel because they do not have a uniform charge. So what we do, we add SDS and SDS if their amino acids are like this. SDS binds with the peptide bond and gives a uniform negative charge to your proteins. So this is the structure of your SDS molecule. Okay, in these conditions, it has this O negative and Na positive is ionized. So this negative charge due to these O's, okay, give a uniform negative charge to your protein molecule. So to each 2 amino acids, 1 SDS binds. So you know that when we acid the protein, it's a 3-dimensional structure, and this SDS helps in breaking all these bonds. That makes this tertiary structure of protein. And then beta mercaptoethanol is a reducing agent. If there are disulfide bridges, it will break these disulfide bridges. Okay, so that at the end you have a linear protein, and your SDS molecules are bound at regular intervals to it, so that they give a uniform negative charge to the protein, right? Now the question arises, why we use two gels, one stacking and one resolving in this, while as in agarose gel electrophoresis we use only one gel. This is because if we make the structure of this gel. The pH of this is 8.8. This is your resolving gel. And this is 6.8. This is your stacking gel. And then you have the buffer tank in which you put this. And the pH of this is 8.3. This is just glycine. So we mix our protein samples with our loading dye. Boil it at 10 degrees so that the protein denatures fully. And then we spin it so the debris settle down, and then the clear supernatant, that's our protein, and the loading dye, we take it and we load it with the help of a pipette into these wells. Depending upon the thickness of the well of the gel, you can make a 1 millimeter thick gel, 1.5 millimeter thick gel. You can make even smaller, thinner gel, like 0.5 millimeter. So thicker the gel, more the volume you can load in your well. So when we apply negative and positive charge, or run it in an electrophoretic tank, the electrophoretic tank of SDS is different from electrophoretic tank of agarose gel. Okay. So what happens here? You have this glycine ions are furnished, plus you have chloride ions. Chloride ions, because this gel is made up of tris HCl. Okay, the chloride ions are furnished from that chloride, right? Chloride ion have a negative charge. Protein have a negative charge because of SDS that we have added to it. And glycine also has a negative charge. But glycine behaves differently at different pH. If we draw the structure of glycine, at pH 8.8, glycine is negatively charged. Okay, as the pH decreases to 6.8, its negativity decreases. So at lower pH, say 6.8 or lower, so it becomes towards more. At pH say 2, it becomes more positive. At higher pH, it becomes more negative. In between, it becomes a what we call as zither ion when the charges are balanced. So at pH 8.3, that's in the buffer. When we run the gel, these glycine ions start to move into the gel. Okay, but once they are into the gel, their mobility decreases because now there's a change in the pH. The pH is 6.8, and at lower pH, the negativity of glycine ions decrease. So they are dragged back. Okay. So chloride ions are faster. So you have chloride ions here. Okay. And then you have your protein. and then you have glycine ions. Now, chloride ions are also not that fast at this lower pH. So what happens? Why we use 6.8 pH? Because if we do not use a stacking gel, and directly the protein are run on a resolving gel. So that means the proteins that are here, this is around say 3 to 4 millimeters, length of the well is 3 to 4 millimeters. So the proteins that are here will enter the well first, enter the gel first, and the proteins that are behind will enter the gel late. Till the protein that is here enters the gel, this protein would have reached here. Okay, that means you're starting a race that's biased, because some people you are leaving earlier. Okay, go start. Okay, and some people you say, okay, now you run. So that's a biased race. So that this race is kind of everyone starts equally, we use this stacking gel. So what happens here when the proteins enter here? The chloride ions are at the front, protein molecules are behind chloride ions, and then you have the glycine ions. So they kind of, chloride ions and glycine ions sandwich the proteins. The glycine ions do not let them move faster, and the chloride ions are pushing them. So they are stacked together in the form of a disc. And when the protein reaches here, it's like this. The whole protein is stacked in form of a disc, and they are at equal position. And now when the race starts, now we can say the race is correct. It's not biased. So once they enter the pH 8.8 resolving gel, okay, the chloride ions move very fast and kind of they clear the path for the proteins. And then glycine ions gets more negative, and it also moves ahead of proteins. Okay, now your protein is left behind, and slowly it can resolve according to its molecular weight. The bigger the protein, it will be difficult for that protein to move through the matrix, the gel matrix. The smaller the protein, it will be easier for it to traverse through the gel matrix, and it will move faster. So at the end of the day, when you see the protein bands, you will find the smaller proteins are moved further, and the larger proteins are kept back. Okay, their mobility is less, right? Now, once you run the gel, okay, take it out from the electrophoretic apparatus, okay, and with the help of the spatula, you put that spatula in between the gels, in between the glass plates, and slowly kind of tilt it so that these glass plates will separate, and on one of the glass plates your gel will be stuck. Okay, then you can slowly, with this help of a spatula, we do not require this stacking gel now. We'll simply cut it out, okay, and mark the gel so that we know which side is which. Now you have a gel and nothing is visible because you have not stained the gel. We do not add ethyl bromide here, okay, because it does not interact with the proteins. So we have to stain the proteins in a different way. Here we use Kumasi Brilliant Blue, okay, that stains the proteins blue. So what we do, we put this gel in a staining solution, okay, that comprises of ethanol, okay, plus glacial acetic acid, okay, water, distilled water, and then you have Comessive Brilliant Blue R250. So you put the gel in this solution, okay, for a while, say 20 minutes, 30 minutes, in 1 hour. Okay, then you keep it on a rocker so that it rocks and the Comessive Brilliant Blue reaches every protein. So after that, you will see that whole of gel has become blue. You'll not be able to see your protein bands. Then there's a process we call as destaining. Then after staining, we destain the excessive staining solution, okay, with the help of a destaining solution that comprises of ethanol and glycol acetic acid and water. So you put it for a while, okay, till you see that your bands are blue, but the gel is water colored. And then when you see it, you'll be able to see it under a white light illuminator, or also on a white sheet of paper. But usually we can use gel dock also to image this, and you will see blue bands of proteins separated according to their molecular weight. The precautions you have to take is that acrylamide and bisacrylamide are again very toxic, and in unpolymerized form they are highly carcinogenic. But once they polymerize, they are safe. Okay, but still you should keep you should wear gloves, you should wear your aprons, you should wear a face mask, so that if there are any spills, you won't damage your eyes. And even if the gel solidifies, still there is an unsolidified or unpolymerized acrylamide bisacrylamide that is kind of associated with that gel. So you should not handle this gel with bare hands and should discard it properly, right? So this was all about your SDS page. If you have any questions, I'm ready to take them.